Hello world, and welcome to another episode of Here we talk about exciting subjects in science, medicine, robotics, astronomy, futuristic ideas, music, or anything interesting in the news. So let's get into it. Researchers successfully created a synthetic mouse embryo without an egg or sperm. How far we have come, because I've learned the normal process for the first steps of human life could only happen by an egg fertilized by sperm. However, today's technology moving fast as it is, we have scientists around the globe that has accomplished this difficult task. They created an embryo without an egg or sperm. Let me repeat that. They created an embryo without an egg or sperm. So, do you ask yourself, what does this mean? Regardless of how you and I feel, this is something that is happening. This is very beneficial. Research about organs in developing stages with an embryo would benefit the human race with the possibility to grow and replace transplantable tissues and organs. The future looks very bright. How can you not love science? So the article we are reviewing today, the headlines read, without egg, sperm, or womb, synthetic embryo models may enable growing organs for transplantation. This article is by the Weizmann Institute of Science. Now, the first paragraph here states that an egg meets a sperm. That's a necessary first steps of life's beginning. Embryonic development research is also common first step. However, in the new study published on August 1st in the journal Cell, researchers from the Weizmann Institute of Science have grown synthetic embryo models of meats outside the womb by starting solely with stem cells cultured in a petri dish. That means that they grown without the use of fertilized eggs. This method opens new horizons for studying how stem cells form various organs in the development of the embryo. It may also one day make it possible to grow tissues and organs for transplantation using synthetic embryo models. So how do you feel about that? Let me know in the comments. Now I'm playing a video for you. The video will be 57 seconds and it's a repeat of letting you know the development of the embryo model on day 8 of its development. It has a beating heart, a yolk site, a placenta, and emerging blood circulation. Professor Jacob Hanna of Weizmann Molecular Genetic Department states that the embryo is the best organ-making machine and the best 3D bioprinter. He goes on to explain that scientists already know how to restore mature cells to stemness. And you ask, what is stemness? If you look up stemness, we will see it's undifferentiated biological cells that can differentiate into specialized cells. In multicellular organisms, stem cells are undifferentiated or partially differentiated cells that can differentiate into various types of cells and proliferate indefinitely to produce more of the same stem cells. They are the earliest type of cell in a cell's lineage. They are found in both embryonic and adult organisms, but they have slightly different properties in each. Now we know what that means. He goes on to explain that until now, in most studies, the specialized cells were often either hard to produce or aberrant, and they tended to form a mismatch instead of well-structured tissue suitable for transplantation. He goes on to say, we managed to overcome these hurdles by unleashing a self-organization potential encoded in the stem cells. On 2 previous advances in his lab, one was an efficient method for reprogramming stem cells back to its naive state. That is, to the earliest stage when they have the greatest potential to specialize into different cell types. The other described a scientific paper in Nature in March of 2021 was the electronic control device the team had developed over 7 years of trial and error for growing natural mouse embryos outside the womb. The device keeps the embryos bathed in a nutrient solution inside of beakers that moves continuously, stimulating the way the nutrients are supplied by material blood flow to the placenta, and closely controls oxygen exchange and atmospheric pressure. In the earlier research, the team had successfully used this device to grow natural mouse embryos from day 5 to 11. The video playing right now is labeled Synthetic Mouse Embryo Models Were Grown Outside the Womb. You can find that on YouTube. It states that continuously moving beakers stimulating the natural nutrient supply while oxygen exchange and atmospheric pressure are tightly controlled. In the new study, the team set out to grow a synthetic embryo model solely from naive mouse stem cells that have been cultured for years in a petri dish, dispensing the need for starting a fertilized egg. This approach is extremely valuable because it could, to a large extent, bypass the technical and ethical issues involved in the use of natural embryos in research and biotechnology. Even in the case of meat, certain experiments are currently unfeasible because they require thousands of embryos, whereas access to models derived from mouse embryonic cells, which grow in lab incubators by millions, is virtually unlimited. Before placing the stem cells into the device, the researchers separated them into 3 groups. One, which contains cells intended to develop into embryonic organs themselves. The cells were left as they were. Cells in the other 2 groups were pre-treated for only 48 hours to overexpress one of 2 types of genes, master regulators of either of the placenta or the yolk sac. We gave these 2 groups of cells a transition push to give rise to extra embryonic tissue that sustain the developing embryo. Now you see in the image development of the synthetic embryo models from day 1 in the top left corner to day 8 in the bottom right. All their early organ progenitors have formed, including the beating heart and emerging blood circulation, a brain, a neural tube, and an intestinal tract. Credit goes out to Weizmann Institute of Science for that. When compared to natural mouse embryos, the synthetic models displayed a 95% similarity in both the shape of internal structures and the gene expression patterns of all different cells. The organs seen in the models gave indication to be functional. Professor Jacob Hanna stated that our next challenge is to understand how stem cells know what to do, how they self-assemble into organs and find their way into their assigned spots inside the embryo. And because our system, unlike a womb, is transparent, it may prove useful for modeling birth and implementation defects of human embryos. Down below, here's a picture that I will leave you with, a diagram showing the innovative method from growing synthetic mouse embryo models from stem cells without egg, sperm, or womb, developed in the laboratory of Professor Jacob Hanna, credited to the Weizmann Institute of Science. So as you look at this, let me know your comments. Leave your comments, let me know.